So in this video, we're going to talk about an introduction to microbiology, history, and nomenclature. So the first question I have for you is that advertisements tell you that bacteria and viruses are all over your home and that you need to buy antibacterial cleaning products. And the question is, should you? And so what I want you to do is I want you to think about this, and we will have a discussion about this in Zoom. So start thinking about whether or not you think we should use antibacterial cleaning products all over your home. Are there maybe instances in which you would want to use those? Are there reasons not to, etc.? So we are going to start by talking about types of different microorganisms. And microbiology is a specialized area of biology that deals with living things too small to be seen without magnification. And so a microbe or a microorganism, again, would be things that are too small to see with the naked eye. So some examples of this would be bacteria, archaea, protozoans, fungi, helminths, viruses, and algae. So what you're looking at in this picture is this brown here. That is going to be human hair. And so to give you an idea about comparison, what you're looking at is that this big pink looking thing here, this is the head of a type of helminth that you guys will learn about in lab that is referred to as Taniosolium. Taniosolium is a pork tapeworm. And you can see it has these little hooklets which allow it to grab onto the lining of the intestine. And so if we compare the head of this helminth to other types of microbes, what you'll see is here we have fungus. So in this case, we're looking at Aspergillus, and we will again talk about this in our lab about fungi. And so we have this Aspergillus, and so this is the size relative to the helminth. And so we have our protozoans. So here's an example of a protozoan. We have a red blood cell. So this is the rough size of a red blood cell relative to these others. And then if you look here, this blue is now the fungi blown up. And what you can see is that relative to this fungus, the bacteria is much, much smaller. So E. coli, for example, is going to be approximately 1/2 micrometers in size. So it's very, very small. We are going to talk about archaea. And you'll notice that viruses are even smaller. Viruses, you'll learn, are acellular, meaning they're not made of a cell, and they are ultra microscopic. You can't even see them with a compound light microscope. And so what we're going to look at now is we are going to look at what do these different groups of organisms have in common? What do they have as differences? So microbes have shaped the development of the Earth's habitat for billions of years. The Earth is approximately 4.6 billion years ago. And so that is when the Earth began. Now, in terms of the first living organism, the first living organism was believed to have appeared on this planet about 3.8 billion years ago. And so that would be our first sign of life. Now, that first sign of life is believed to have been single-celled. It was a unicellular organism. From that last common ancestor, we have branched off into 3 different cell types. So we have our eukaryotic cells. Eukaryotic is referring to true nucleus. Those are organisms that have a membrane-bound nucleus that surrounds their DNA. And then the other 2 categories are going to be archaea and bacteria. Both of those types of organisms are single-celled. They have no true nucleus. They are what we call prokaryotic cells. But archaea and bacteria are still distinct from one another. And in fact, archaea are actually believed to be more closely related to eukaryotic cells than they are to bacteria. So we are going to go through and talk about the different characteristics for these different organisms. And so we're going to start with bacteria. So bacteria are prokaryotic. Again, that means these are cells that lack a membrane-bound nucleus. So they have no nucleus. They have a cell wall, and that cell wall is usually made of peptidoglycan. Now, what that means, peptido refers to protein, glycan refers to sugar. So its cell wall is going to be a mixture of protein and sugar. You're going to see that that is distinct from many other types of cell walls. In eukaryotic cells, for example, the cell walls are typically what we call polysaccharide. There are many sugars linked together. But in bacteria, it's a mixture of protein and sugar. And so that makes it unique from eukaryotic cells. Bacteria exist being unicellular. Uni, think of like a unicycle, has one wheel. Unicellular means one cell. So they exist as one single cell. In terms of their reproduction, they reproduce by a process called binary fission. And what that means is that this is a type of reproduction that is asexual. A means without sex, meaning without a partner. For bacteria, they are simply going to just make a copy and divide. And so that is asexual reproduction. And so bacteria are going to reproduce asexually. In terms of their DNA, you're going to learn is their nucleic acids, meaning that that is the way that organisms store genetic information. In the case of bacteria, their DNA is going to be circular. So while our DNA is linear, meaning they're long strands, bacterial DNA is going to be circular. It's a circular piece of DNA. Now, in terms of how bacteria get energy that they need, all organisms have to be able to take in energy. So in the case of bacteria, some bacteria are photosynthetic. What that means is that they do photosynthesis, and they are what we call autotrophic. Auto means self. Trophic is referring to feeder. Self-feeder. These are organisms that are able to make their own food. And so cyanobacteria, which is a type of bacteria found in water, is an organism that can do photosynthesis, just like plants. They can take carbon dioxide, water, and sunlight and convert that into chemical energy in the form of sugar. So they're self-feeders. They can make their own sugar. Other types of bacteria are heterotrophic. Hetero refers to other, trophic is feeder. So many bacteria are going to be heterotrophic. They have to consume food in order to survive. They're not able to simply just take in and make their own food. Next, we have archaea, and archaea are prokaryotic, which means again that they also lack a membrane-bound nucleus, so they do not have a nucleus. Their cell wall lacks peptidoglycan. So they do have a cell wall, however, it is not peptidoglycan. It is something that's referred to as pseudomurin. So it is like peptidoglycan. It is still protein and sugar mix, but it's not chemically the same as the peptidoglycan. Instead, it's the pseudomurin. Archaea are unicellular. Again, they reproduce by binary fission, which again, remember, is a type of asexual reproduction. They also have circular DNA, just like bacteria do. One of the interesting things about archaea is that they tend to be, not always, but they tend to be what we call extremophiles. Extreme, like it sounds, and then phile is lover. So they tend to love extreme environments, meaning they grow best in these very extreme environments. So if we think about a thermophile, what would you think a thermophile likes? Well, the answer is a thermophile likes heat. These are our heat lovers. If we talk about halophiles, halo is referring to salt. So if you look over on the right, you can see this salt crystal and it has this pink hue on it. And that pink hue is a type of organism that is called Halobacterium. It's not actually a bacteria. It's actually an archaea, but it can grow in extreme salinity, meaning high salt environments. Most organisms can't tolerate high salt, yet this organism can, and it can grow on these salt crystals, and so we would call this a halophile or a salt lover. So next we have our methanogens. These are going to be our organisms that produce methane, which is a gas, as a waste product of respiration. And so this is just a little bit of information about archaea. So now we're going to move on to our eukaryotic organisms, meaning these are organisms that the cells are made up of cells that have a nucleus. They have this membrane-bound nucleus. So the first one, fungi. Fungi are eukaryotic. So again, their cells have a membrane-bound nucleus. Cell wall is made of something called chitin. Chitin is a polysaccharide. It's many sugars linked together. So chitin, again, polysaccharide made of many sugars. Fungi are heterotrophic. They have to consume their food. So if you think of things like mold, where do you typically think of mold as growing? Dark, damp places, right? And so what that tells you is that those organisms are not likely to be photosynthetic. They're not likely to do photosynthesis because they grow in the absence of sunlight. In the case of fungi, while they're heterotrophic, many are what we refer to as a saprobe. It feeds on dead, decaying matter. So next, if we look at the way that the cells are arranged, they can be unicellular, meaning they exist as one single cell. And an example of this would be yeast. So at the top here, we have a type of yeast that is called Saccharomyces cerevisiae. Saccharomyces cerevisiae, the name literally means sugar fungus that makes beer. This is a type of yeast that is used to make beer, bread, and wine. And so it's able to do what's called fermentation. And so this would be an example of a unicellular fungus, so a yeast. Fungi can also be multicellular in the case of molds and mushrooms. So we have our bread mold, for example. We have our mushrooms. These would be examples of multicellular types of fungi, meaning that they're made of more than one cell. In terms of their reproduction, they can reproduce sexually or asexually. So either with a partner or without. So if we look at this bread mold, for example, it has these types of spores that are used for reproduction. And in this case, we're looking at what are called sporangiospores. Notice that we have these sporangium. These are these structures that contain the spores, and you'll see these little filaments coming off of them. Those filaments are referred to as hyphae. You can tell by looking at this that this is asexual reproduction. It doesn't involve a partner because only one hyphae is going to the structure. So this is going to be something that's used for asexual reproduction. But fungi can do both. They can do asexual or sexually. In terms of their DNA, they have linear DNA, so long strands of DNA versus circular. And so their DNA is going to be these long strands in contrast to bacteria, which have circular chromosomes. So next we're going to move on to our protozoans. And protozoans are eukaryotic. Again, that means that they have a membrane-bound nucleus that surrounds the DNA. These will usually lack cell walls. So protozoans are thought to be more kind of animal-like. And so in this case, these are going to lack cell walls. They are usually heterotrophic. So again, they have to usually consume food in order to survive. Protozoans are going to be unicellular. So they exist as one single cell. You can see on the right here, this top picture in the yellow, that is amoeba, and it moves by the pseudopods. You can see it moves by pseudopods. Basically, it does a crawling motion, and it can also use pseudopodia to engulf its food. So they are going to be unicellular. They exist as one single cell. They can reproduce either sexually or asexually. So again, with a partner or without. And in terms of their movement, again, they could move by pseudopods. They can move by flagella, so these long tail-like extensions. They could move by cilia, which are these tiny hair-like projections that cover the whole surface. So a paramecium, for example, would move using cilia. And then some are going to be non-modal, meaning they're not able to move. What you're seeing in the bottom here, these little purple squigglies that you're seeing that are mixed in with the red blood cells, That is a type of organism called trypanosoma. And trypanosoma is an organism that can cause what's called African sleeping sickness. And African sleeping sickness, like the name suggests, what ends up happening to patients is that when they have this infection, and this infection is heavy, meaning there's a lot of these organisms in the blood, the organisms are then taking the host's food. So if this organism lives in your bloodstream, normally the way that your body gets food is that you eat your food, you digest it, the molecules become small enough, they diffuse from your intestines into your bloodstream, and then the food is going to circulate through your bloodstream to the other tissues in your body where that food is going to be used to make ATP. This organism lives in the blood. It can exist in the blood. And so what ends up happening is that when it lives in the blood, it's utilizing your food. If it's utilizing your food and you aren't, what ends up happening is that your cells are unable to make ATP. They're unable to make energy. And so this is why people who have this disease, they end up basically going into a coma because their cells don't get the glucose that they need, and therefore they don't make the ATP, and the brain cells stop functioning. And so the interesting thing about this particular organism, because most organisms can't survive in the bloodstream, because if you think about it, in your bloodstream are your white blood cells, which are basically your immune cells. Those are the cells that fight off foreign invaders. So most organisms, when they get into your bloodstream, they're bombarded by white blood cells, which are there to destroy them. However, trypanosoma has a unique adaptation in that it does something called antigenic switches. It basically is able to change its antigens. An antigen is a molecule on the surface of the organism that would normally elicit an immune response, meaning it would cause the immune system to respond because it would recognize it as being foreign. In this particular organism, what it does is it has a particular set of antigens. We'll call it antigen A. The immune system will recognize antigen A as being foreign, and the immune system will mount an immune response. They'll make antibodies to antigen A, but trypanosoma's tricky. It mutates. It changes its antigens. So now antigen A is not on the surface, but now it's making antigen B. So those antibodies that recognize A, those antigens are no longer on the surface. Now the organism has antigen B on its surface. And then it's going to take time for the immune system to recognize it and to respond. And by the time the immune system responds, it changes its antigens again. So it's almost like it has its disguise, and it's able to take off its disguise, and now the next layer is exposed, and so on and so forth. And so this is what allows trypanosoma to be able to live within the bloodstream. So now we're going to move on to our algae. Algae are eukaryotic. They have a cell wall with cellulose. Just like chitin is a polysaccharide, cellulose is also a polysaccharide. So many sugars link together, just like the cell wall of fungi is made of polysaccharide. Algae are photosynthetic. They're able to do photosynthesis. They exist as unicellular or multicellular forms, so it could exist as either. They can reproduce sexually or asexually, so they can reproduce with a partner or without. And for algae, they often contain pigments which make them appear green, red, or brown. Brown algae, think of what we typically would refer to as seaweed, the seaweed that washes up on the beaches. That is a type of algae that's called brown algae. It's a type of brown algae. Now, because they are photosynthetic, we should also write that they are also autotrophic. They are self-feeders. So self-feeders that are able to make their own food. Now, we're going to move on to our viruses. Now, viruses are a little bit different than everything else we've talked about. And that's because viruses are acellular, meaning they are not made of cells. So again, A means without, so acellular, without cells. So because they are acellular and they're not made of cells, cells are the fundamental unit of life. And so if they're not made of cells, they're not living. They're considered to be non-living. And that's because viruses are what we call obligate intracellular parasites. Obligate means that it has to be within the cell. So viruses cannot reproduce without a host cell. And so the virus has to get into a host cell in order for it to reproduce. So like the coronavirus right now, that virus has to infect only particular types of cells, only cells that have the appropriate receptor, like the cells in the respiratory tract, for example. And so viruses have to be able to get in and infect a host cell in order for it to reproduce. This is why it's non-living. In terms of its genetic information, DNA or RNA, but not both. Now, this is different because our cells and all of the other organisms that I had talked about previously, they store their genetic information as DNA. We store information as DNA, and RNA is used kind of as the messenger. But DNA is the way that we store it. Viruses, on the other hand, can store their genetic information as RNA. For example, coronavirus, right? The one that's causing COVID, that is a RNA-based virus. Influenza is an RNA-based virus. So they have a different type of genetic makeup. They don't store genetic information as DNA. Instead, they store it as RNA. Now, in terms of viruses, viruses can be enveloped or naked. Enveloped basically means that it has an envelope, which is a structure that is made of a cell membrane. These are animal viruses, and the way that they get that envelope is when they bud off of the animal cell, they take some of the animal cell membrane with it. Now, the advantage that gives the virus is that it helps it to evade the immune system because the outside of the virus looks very much similar to the host cells. And so it's more difficult for the host to respond because it's having a more difficult time recognizing that the virus is within the cells. We have a capsid. A capsid is a protein coat. So viruses are very simple structurally. They're typically going to be genetic information. So again, DNA or RNA. And then that is going to be surrounded by this protein coat, this capsid. And so viruses lack a lot of, or all, cellular machinery that living cells have. And so this is what makes viruses have to be obligate intracellular parasites because they lack those cell machinery. They have nucleic acid, they have this protein coat, but that's pretty much about it. In terms of DNA or RNA, notice it says or. So it's usually one or the other, but not both. Now, there have been examples of viruses that can have both, but it's extremely rare. That would be an exception to the rule. One of the things that you'll learn about in biology, while we can say, for example, bacteria, most have cell walls made of peptidoglycan. Not all have a cell wall of peptidoglycan. There are always exceptions to the rule. There are viruses, for example, mycoplasma we'll talk about later. It lacks a cell wall, doesn't have one, but most other bacteria do. And so one of the things you'll learn about biology is while we have a rule that usually this is the case, like viruses, usually the genetic information is DNA or RNA, but not both. Again, there's always going to be exceptions to this rule. And then we get to our multicellular animal parasites. A helminth, for example. A helminth is a worm. So we will talk about this a lot in lab. We'll talk about different types of worms, but we have our flatworms, like our tapeworms, for example, that's here in the bottom. We have our roundworms, like heartworm, for example. And so these would be our helminths. Because they are made of animal cells, they're animals, right? These worms are animals. Their cells lack a cell wall, so they don't have that rigid cell wall. They are heterotrophic. They're other feeders. Animals have to feed in order to survive. We can't make our own food. These organisms could reproduce sexually or asexually, so with a partner or without. And the reason that we're studying these, while these worms are actually quite large, they do have microscopic stages. So they have these microscopic stages and they can cause disease. And so this is why we will be talking about these in microbiology as well. So, question for you. Which of the following is not a characteristic of bacteria? Is it red? They are prokaryotic. Yellow, have peptidoglycan cell walls. Green are multicellular. Blue, reproduce by binary fission. Purple, have the ability to move. And when I say have the ability to move, I'm referring to as a group. Some can be modal. And so that's what that's asking. So I want you to pause your video. Think of your answer, and when you are ready, push play and you can hear the answer. So, which of those statements is not true of bacteria? The answer is going to be green. They are multicellular. They are not multicellular. They are unicellular. They exist as one single cell. And so the statement saying that they're multicellular is not true for bacteria. Bacteria are prokaryotic. They are made of cells that lack a membrane-bound nucleus. They have peptidoglycan, usually, in their cell walls. They can reproduce by binary fission, which again is an asexual type of reproduction. And they have the ability to move. Do they all move? No, they're not all modal, but they do as a group have the ability to move. So this next part is something that you are going to do in the discussion. We are going to compare and contrast the various types of organisms. So for bacteria, you would fill this in. Are they prokaryotic or eukaryotic? So you want to go back through your notes and fill in if that's prokaryotic or eukaryotic. For bacteria, what is the composition of their cell wall? What is their cell wall made of? Or for certain organisms, do they lack a cell wall? Are they heterotrophic or autotrophic or both? Meaning, can they be both heterotrophic or autotrophic? Are they unicellular or multicellular or both? Meaning they could be either or asexual reproduction or sexual reproduction or both. So I want you to take some time and fill in this table by yourself first, and then you will discuss it with a group of people in a Zoom meeting, and we will talk about and fill in this table together. But you should work on this on your own first, and then you will discuss it with a group. So now we're going to move on and talk about microbes in our lives. How do microorganisms affect our everyday lives? So microbes are very easy and very difficult to study. And that is because of multiple factors. One is that they reproduce rapidly. So we can grow bacteria very quickly in culture. Bacteria like E. coli, for example, reproduce every 20 minutes. So that makes them more on the easy side in order to study. They can be grown in large populations in the laboratory, again, because they reproduce and because they don't take up much space. We can culture large amounts of these organisms. But what makes them difficult is that they can't be seen directly, right? Because they're microbes, they're not visible with the naked eye. You have to use a microscope, for example, to see them. So they are usually analyzed through indirect means. So either using a microscope or doing what we call biochemical tests to study different aspects of their metabolism, etc. And so this is what makes microbes both easy and difficult. It has pros, it has cons. Now, microbes are ubiquitous. And what that means is they're found almost everywhere in nature. So where can they be found? Well, they can be found in very diverse environments. They could be found deep in the Earth's crust. They can be found in polar ice caps and in oceans. They can be found inside the bodies of plants and animals. They can be present in the Earth's landscape, and they are essential to life. And in fact, if we read about microbiomes, so the types of bacteria, for example, that are found in our body, what you would find is that our bodies do not function properly without the help of those microbes. Those microbes help our body in a variety of ways, whether it be digestion, etc. And so we need those microbes. They are absolutely essential to life. Now, remember that I said that some bacteria, for example, are photosynthetic. They are able to convert solar energy to chemical energy. And so this is going to be the light-fueled conversion, so using sunlight, and converting carbon dioxide to organic material, so glucose, for example. And this can be accompanied by the formation of oxygen, meaning that plants, for example, or cyanobacteria, for example, when they do photosynthesis, they're going to produce oxygen as a byproduct. Us as animals, we need that oxygen to breathe in because we need oxygen to do cellular respiration. And so this oxygen that we breathe in comes from organisms that can do photosynthesis. Now, anoxygenic photosynthesis occurred in bacteria before plants evolved. And anoxygenic means without oxygen. They do not produce oxygen as a byproduct. This is going to be more efficient, however, in extracting energy from sunlight. But again, it doesn't produce oxygen to be put back into the atmosphere. Oxygenic photosynthesis is evolved from anoxygenic photosynthesis. So this is a later form of photosynthesis where basically, again, they're going to produce oxygen. And the photosynthetic microorganisms are responsible for about 70% of the Earth's photosynthesis. So while we often think of plants as being the major contributor to oxygen in the atmosphere, in fact, it's actually photosynthetic microbes that produce a variety or the biggest range or the biggest amount of oxygen in the atmosphere. So microorganisms are the main forces that drive the structure and the content of soil, water, and the atmosphere. For example, microbes can produce carbon dioxide, NO gas, we have methane, CH3, and these gases are used to help insulate the Earth's atmosphere. Microorganisms are the most abundant cellular organisms in the oceans. Now, notice it doesn't just say the most abundant thing in the ocean. Viruses are actually more abundant in the ocean than bacteria, but bacteria would be the most abundant cellular organisms because remember that viruses are not made of cells. So viruses are the most abundant inhabitant of the ocean. Bacteria and fungi live in close association with plants and assist them in obtaining nutrients, water, and also helps to protect them against disease. And so microbes really play an important role in shaping our planet. So some historical uses of microbes by humans. We can use microbes to produce bread, alcohol, right? So remember I referred to Saccharomyces cerevisiae, baker's or brewer's yeast. They can do fermentation and they produce alcohol and carbon dioxide, which is why yeast is used in bread. It's used to help the dough rise. Microbes can be used to make cheese, for example, blue cheese, brie cheese, etc. Microbes can be used to help with treatments of wounds and lesions. They can be used to produce antibiotics, for example. They can be used to mine precious metals. They can be used to clean up human-created contamination. So like an oil spill, for example, there are microbes that naturally can consume the oil that spills into water. And so this bacteria can be put into the water and it can naturally digest and break up the oil that spilled into the water. So when we think about microbes, we often need to think about that they play a role in biotechnology. And so there are several ways that they can play a role in biotechnology. One is through what's called genetic engineering. This is going to manipulate the genetics of the microbes, plants, and animals for the purpose of creating new products and genetically modified organisms. And so they can be used to basically change the DNA of a plant, for example, to make that plant to be more resistant to a particular pest. And so that would be our genetically modified organisms. We have recombinant DNA technology, which makes it possible to transfer genetic material from one organism to another and deliberately alter the DNA. So, for example, the first human recombinant drug to be produced was something that was called humulin. This was produced way back in the 80s, and humulin is human insulin. And so for patients who are diabetic, for example, if they have type 1 diabetes, which is juvenile diabetes, what happens in those patients is that they have an autoimmune disease. Their immune system is going to attack their own cells. And specifically in type 1 diabetes, their autoimmune disease is actually targeting their pancreatic beta cells. It's a type of cell in the pancreas. Those pancreatic beta cells are the cells that are responsible for producing insulin. Insulin is important in your body because when you eat food and your blood sugar goes up, right? Because as you start to digest your food, it's going to be absorbed into your bloodstream. When it goes into your blood sugar is going to rise. In response to that, your pancreas is going to secrete, it's going to produce insulin, and it's going to release insulin into the bloodstream. Insulin is the signal that tells the cells in your body, hey, sugar is available. Start to take in the sugar into the cells so that the cells can use that sugar to make ATP. So insulin is essential to basically being able to get glucose into the cells. Without it, glucose can't be transported into the cells. So people who are diabetic, they have their pancreatic beta cells that are destroyed. So their body is no longer producing insulin. And so when they eat sugar, for example, and their blood sugar starts to rise, they don't have that signal that tells their cells to take up glucose. And instead, the glucose stays in the bloodstream. And so the solution to that is to give these patients injections of insulin when they eat food to help regulate their blood sugar. Now, historically, where that insulin had came from previously, usually insulin came from one of two places. One, it could be isolated from cadavers. So people who had died, they could get insulin from. Or it could be used from animals. So they could use pig insulin or cattle insulin or other types of animals to produce insulin. The problem with that approach is that it's not human insulin. And so some people had adverse reactions to this not normal insulin protein. It's a different protein than humans make. So the idea then was to take the human insulin gene, so the gene, the DNA sequence for human insulin, and you can take that gene and put it into bacteria, and you're going to learn that because the genetic code is nearly universal, you can take a human DNA sequence and put it into bacteria, and then bacteria will read that DNA sequence and it will make the appropriate protein. And so this was used to make this humulin, this human insulin. They took the human insulin gene, put it into bacteria. They could grow the bacteria in these large bioreactors, these very large containers, and they could produce lots of insulin very quickly. And that insulin was human insulin because it was from the human sequence, the human gene for human insulin. And so that is a type of what we call recombinant DNA technology. We can have bioremediation. This is going to use microbes that are already present or introduced intentionally to restore the stability or cleanup of toxic pollutants. So again, just like the example of the oil spill. Now, while we often think of bacteria, we usually have a negative connotation that goes with it, meaning we think about them causing disease. But what we really need to realize is that the vast majority of microorganisms that are associated with humans are either harmless, meaning they have no effect, or in fact, they are beneficial. And so one of the big things that we've started to learn is that, in fact, bacteria actually are helpful to us. And if we get a disruption in what we call our microbiome, or the types of bacteria that are living in our body, there are lots of diseases associated with that. And so, in fact, these bacteria in many cases are actually beneficial. However, there are some that cause disease. And if they are a disease-causing organism, we refer to that as a pathogen. So a pathogen is a microbe that causes disease. There are over 2,000 different microbes that cause disease, and you're going to learn a lot about those throughout this class. 10 billion infections occur across the world every year. Infectious diseases are among the most common cause of death worldwide. Now, what you'll notice is that in the US, it's not a top form of, or not a top cause of death. But around the world, in fact, infectious diseases are definitely still a big problem, and they are a leading cause of death. So what you're looking at here, this is comparing the top causes of death in the United States versus worldwide. So you do not have to memorize this table. It was just to give you an idea of the differences that the US faces versus the rest of the world. Now, if they're colored blue, blue indicates that it's not currently known to have an infectious cause, meaning that these are things that are causes of death that are not related to infection. Notice if you look at the top causes of death, so in the US, the top cause of death is going to be heart disease. There's not really an infectious component to that. It has more to do with diet, lifestyle, genetics, etc. Second leading cause of death would be cancer, followed by chronic lower respiratory diseases, so COPD, so lung diseases, etc. Accidents would be number 4, stroke would be number 5, Alzheimer's disease is number 6, diabetes is number 7, that was the one I just explained to you. Number 8, now we finally see the first infectious cause of disease in the United States. So now we have our influenza and our pneumonia. Those are infectious causes of disease. And then we have kidney disease followed by suicide. So that one always surprised me, that the number 10 cause of death in the US is actually suicide. That's pretty crazy. If we compare that to worldwide, most common cause of death is still heart disease. So that is still number 1. In the world, the second highest cause of death would be stroke. 3 would be COPD, so a lung disease. Number 4, lower respiratory infections. So notice that again, this is going to be associated with a microbe. Number 5, respiratory cancers. Number 6, HIV and AIDS, which is caused by a virus. So again, that's the infectious cause. Diarrheal diseases is number 7. So in places that have poor sanitation or not clean water, diarrheal diseases can be very fatal. And so that is actually number 7. That is an infectious cause of disease. We have diabetes, then we have road injury, followed by hypertension, which is basically high blood pressure. And so you'll notice that if you look in the world, 3 of the top 10 are infectious. In the United States, however, only 1 of the top 10 is infectious. And in fact, it's only number 8 compared to worldwide. It was number 4, 6, and 7. And so there's this disparity basically in how deadly infectious diseases are. If you think about the US, we typically have we have good sanitation, we have clean water, we have good access to medical care. And that really is what kind of shapes that the US typically doesn't have this high cause of death due to these infections. So there are many microbes and diseases that are either emerging or re-emerging diseases. COVID, that is the perfect example of an emerging disease. We have other coronaviruses that have caused disease. SARS, for example. We have MERS, which is Middle Eastern Respiratory Syndrome. So we have other types of coronaviruses that have caused disease, but we have this new coronavirus, which causes us to have COVID, for example. We have AIDS, there's hepatitis C, Zika virus, which is associated with mosquitoes. We have West Nile virus. We have tuberculosis. And so what we see is that we often are going to see these either brand new diseases showing up or reemerging, things that were pretty much gone for a while, but then restarted to show up again. Think of when we had a measles outbreak, for example. There have been found to have associations between non-infectious diseases and microbes. And so again, this comes down to this human microbiome. And that if you get a disruption to that, if you get this disruption in the types of microbes that are found in the body, certain diseases can occur. And they're starting to see more of an association between infectious agents and diseases that were previously thought to not have an infectious component. An example of this would be gastric ulcers. So having an ulcer in your stomach historically wasn't thought to be infectious. It was thought to be a product of diet, for example, of lifestyle, etc. However, we now know that there is a possible infectious component to this as well. Helicobacter pylori, H. pylori, you guys will learn more about. H. pylori is a type of bacteria that can be found in the stomach, and it's able to withstand stomach acid, and it can be the infectious cause of ulcers. And so ulcers have now been found to also have an infectious component. Multiple sclerosis, obsessive compulsive disorder, coronary artery disease, obesity, etc. These have all been linked to chronic infections with microbes. So again, diseases that were thought to not be due to anything infectious have now been found to actually have an infectious component to them. So we're starting to see some trends in infectious diseases. And one trend is that we're starting to see an increasing number of patients with weakened defenses. And what ends up happening is when a patient has a compromised immune system, they are then subject to infections by common microbes that are not pathogenic, meaning not disease-causing, to healthy people. And so we call these types of microbes an opportunistic pathogen. It's a microbe that if you are healthy, your immune system will fight it off and you'll have no problem. However, in patients who are immunocompromised, meaning their immune system is weakened, those particular patients are now susceptible to these microbes that normally don't cause disease. And so we're starting to see more of a trend of these diseases that are caused because of weakened immune systems. We also are starting to see a big increase in microbes that are resistant to drugs. Certain types of conditions were once fairly easy to treat. An example of this would be gonorrhea, which is a sexually transmitted infection. This historically was relatively easy to treat. If you got gonorrhea, you took an antibiotic, problem solved. However, we are starting to see multi-drug resistant strains of Neisseria gonorrhoeae, which is the organism that causes gonorrhea. And so what was once an easy to treat sexually transmitted infection is now very difficult to treat because the bacteria has become resistant to many of our antibiotics. So now we're going to move on and talk about naming and classifying microorganisms. So in order to do so, we need a few terms. So the first term is going to be taxonomy. And taxonomy is going to be the science of classifying living things. So basically, how we're going to group different types of microorganisms or different organisms in general. When we talk about nomenclature, nomenclature is the assignment of scientific names to the various taxonomic categories and to the individual organisms. So again, nomenclature, think name. It's naming things. Classification. Classification is the orderly arrangement of the organisms into a hierarchy. You will see that we will talk about classification schemes where we start with domain being the most broad form of classification, working our way all the way down to species, which is going to be the most specific. Identification is the process of discovering and recording traits of organisms so that they can be placed into an overall taxonomic scheme. And so it's important that we figure out what are these characteristics that group different organisms together. So when we talk about nomenclature, again, nomenclature or naming of organisms was established in 1735 by Carolus Linnaeus. And when he named these organisms, the names were Latinized. They're Latin. One of the nice things is you'll start to see trends in the way that organisms are named. And there are things that you can learn about an organism simply by their name. And you'll see this in a minute. So when we look at how organisms are named, they have what's called a binomial nomenclature. Binomial means two names. The first of the two names is going to be the genus. You need to capitalize the genus. The second of the two names is going to be the specific epithet or the species. When we write this, this is going to be lowercase. So genus is capitalized, species is lowercase. Both names are underlined separately. So if you're handwriting the organism name, you will underline it separately, or it's italicized if you type it. Now, if you're handwriting, don't try and handwrite it looking like italics. That doesn't work. If you handwrite it, you should underline genus and species separately. So if you think about humans, for example, and we think about our binomial nomenclature, our two-name name, our name would be referred to as Homo sapiens. And so notice when I wrote it, the genus, the H is going to be capitalized. Species is going to be sapiens. That is going to be lowercase. And notice because I hand wrote it, I underlined the genus and species separately. I don't just draw a line across. I underline individually. So if we look at the way that organisms are named, they may be descriptive, meaning they may tell you something about the organism themselves, or they may have a name that honors the scientist who discovered it. So, for example, Escherichia coli. Escherichia coli honors the discoverer, Theodore Escherichia. So that's the Escherichia. Coli is going to be used to describe the bacterium's habitat. So if you think about coli, what part of the body do you think that refers to? And that is the colon, which is the large intestine. And so E. coli is a type of bacteria found in the gut, and it is part of your normal flora in terms of bacteria that's in your gut. And so the coli, the species name, is going to describe the bacterium's habitat. So again, E. coli is going to be found in the colon or the large intestine. Now, we could also have Staphylococcus aureus. Staphylo refers to what we call the arrangement, meaning the way the organisms are arranged. In this case, they are clustered, what we call staphylo. So staphylo refers to grape-like clusters. The coccus, or cocci, which is plural, is going to refer to the morphology or the shape. This bacteria is spherical or round. So when we look at Staphylococcus aureus and we look at it under the microscope, it's round or spheres, and they're grouped into grape-like clusters. That's their arrangement. So that's the genus. Aureus is used to describe the gold-colored colonies, because if you know the atomic symbol for gold, it's AU. So if you look at Staph aureus growing on a plate, the colonies have this golden yellow color. It produces this pigment that makes the bacteria to appear yellow. And that's where the aureus comes from. It comes from the golden color of that particular bacteria. And so you'll notice that these organisms, their names actually tell you something about that organism, where they're found, what they might look like, who discovered it, etc. So when we write out organism names, the very first time we describe it, let's say in a paragraph, we have to write out the genus and species. You don't abbreviate the very first time. You have to fully write out the genus and the species. However, if you continue to talk about that organism, then you can abbreviate the genus. So let's take a look at this. So if this is what I'm writing, Escherichia coli and Staphylococcus aureus are found in the human body. So notice that they're italicized because they are organism names. And when we write them out the first time, they have to write out the genus and the species. However, if we continue to talk about those organisms, let's say it says E. coli is found in the, so notice that the genus is now abbreviated. We don't have to continue to write out Escherichia coli. So E. coli is found in the, we said colon or intestine. And S. aureus, again, we can abbreviate the genus because we already mentioned it in the sentence before. Staphylococcus aureus is found. Anybody know where Staphylococcus aureus can be found? Answer is skin. It's a type of bacteria that's found on the skin. So we can write S. aureus is found on the skin. So again, first time we talk about it, we need to write out genus and species. When we continue to talk about that organism, we can then abbreviate the genus and simply write out the species. So we can't simply just say, without saying Escherichia coli first, we can't just simply write E. coli because there are more organisms than Escherichia coli. There is a protozoan, for example, that is Entamoeba coli. So if I were to write E. coli, am I talking about Entamoeba coli? Am I talking about Escherichia coli? So the first time you have to write out the genus and the species. After that, then you can abbreviate the genus with the capital letter, period, and then the species in lowercase. So we have, remember, classification, meaning the way that organisms are grouped. And so our current system was proposed in 1978 by Carl Woese. And the way that he classified organisms was different than the way this had been done historically. And what I mean by that is that the way that organisms used to be classified would be simply by looking at the organisms. Okay, well, these organisms look alike, so they must be classified together. And these organisms, there's more diversity, they must be classified separately. But if you think about it, one of the things that if you think about humans, for example, we are all one species, meaning the most specific form of classification. Physically, right, there's a lot of variation between people. So we can't just simply look at things as a good way to classify organisms. And so our current system, this what we call 3-domain system, was classified by looking at ribosomal RNA. And so this ribosomal RNA is a type of RNA that's found in the ribosome, which is an organelle that's used to produce proteins. And so if they compare the sequences of these ribosomal RNAs, the more in common, meaning the more similarities between their sequences, the more closely related they are. The greater the number of differences in their ribosomal RNA, that means that those organisms are more diverse, they're more distinct, and they evolved from each other a lot longer ago in our evolutionary history. And so they can use this ribosomal RNA in order to classify organisms. Now, in our current system, we have a 3-domain system. And our 3-domain system is bacteria, archaea, and eukarya. Bacteria and archaea, remember, are both prokaryotic, meaning that they lack that membrane-bound nucleus, organisms that are in the domain eukarya are eukaryotic, meaning they're made of eukaryotic cells. Now, domain is the most broad form of classification. Then we become more specific. And our next classification is going to be our kingdom. So, broad, our domain is going to be eukarya. Then we can start to group organisms by kingdom. And so historically, we've had this 4-kingdom system: Protists or Protista, fungi, Plantae, plants, Animalia, animals. Those were typically, classically, our 4-kingdom system. However, this system is constantly changing and evolving. And that's because protist, the way that an organism was classified as being a protist, was that it was a eukaryotic cell, but it wasn't a fungus, an animal, or a plant. So it didn't meet the criteria to be any of those 3, but it was still eukaryotic. So they put all of those organisms in Protista. Think about if you have a junk drawer in your house. So like I have a drawer that if, guests are coming over to my house and I'm trying to clean up quickly, I throw stuff in that drawer, right? I put it in there and I say, I'll deal with this later. That was like kingdom Protista. That was scientists' way of saying, we don't know what to do with these organisms. All we know is that they're eukaryotic, but they're not plants, animal, or fungi. So we're going to put them in this other category. We're going to put them in this kingdom Protista, and we'll deal with that later. And so what they found was that a lot of the organisms that were classified as being protists were actually fairly diverse, and that wasn't really a great way to classify those organisms. So that kingdom Protista, which has been around for a while, is now being reclassified, and it's being separated into multiple kingdoms. So you'll probably see over the next, several years that this is a constantly evolving and constantly changing way to classify organisms. It changes all the time. So if we talk about protists, this could be things like slime molds, algae, protozoans, right? Think of algae and protozoans. They are very different from one another. Algae are more plant-like, protozoans are more animal-like. So putting them together, not necessarily the best system, which is why these are being separated now into more kingdoms. If we talk about fungi, so fungi would be your molds, mushrooms, and yeast. Molds and mushrooms, remember, are going to be multicellular. Yeast is going to be unicellular. But they share certain characteristics together, like composition of their cell wall, the fact that they're heterotrophic. Many are saprobes. They feed by dead, decaying matter, etc. So those would be our fungi. Plants, you can think of a lot of different plants. So we have something as simple as a moss. A moss is a very primitive plant. It lacks a vascular system. And so this is why mosses grow low to the ground. They have to get nutrients and they have to get water by simple diffusion. They're not like the other plants. Ferns then evolved to have a vascular system, but they still don't use seeds for reproduction. They use spores. Conifers, so these are our cone-bearing plants. Think of like a pine tree, for example, that would have cones on it. Those plants can grow tall because they have a vascular system, and they now reproduce with seeds. The seeds are contained within the cones. And then lastly, we have our flowering plants, what we call our angiosperms. These are now plants that have evolved to have seeds, but the seeds are now encased within a fruit. And that helps because that fruit attracts animals to eat it. Animals will ingest the seeds, and animals are modal. They'll move to a different area. They will defecate, out comes the seeds, and it helps with seed dispersal. Flowers are an adaptation to help with pollination, right? To have pollinators to help pick up pollen from one plant and put it on another. And so those would be our plants. Animals, you can probably think of 100 different animals that you could put here. And so if I look at different examples of animals, we could have humans, so we are animals, dogs, monkey, octopus, insects, corals, sea stars, sponges. I mean, the list can go on and on. Animals are the last type of classification in terms of kingdom. So if we look at what we call a phylogenetic tree, we're looking at evolutionary relationships, meaning how far ago did these organisms diverge into separate classifications. So if we look at this first ancestral cell line, this first living cell, that cell at some point during its growth acquired a mutation. Mutations are changes in the DNA sequence, and those changes can happen randomly. Well, what can happen is that those mutations, those changes, can then lead to two different groups of organisms. So if we look at the first split, we have this blue and the red. Well, the bacteria split from archaea and eukarya early on. Notice what's interesting is if you look, archaea are more closely related to eukarya than archaea is to bacteria. So even though they are both prokaryotic and even though they are both unicellular, archaea still have more in common with domain eukarya than they do domain bacteria. And so that's why they're shown this way, where they diverged more recently relative to when bacteria split from both domain eukarya and archaea. So if we look at domain bacteria, we have our endospore producing bacteria, Bacillus, Clostridium, etc. We'll learn a lot about endospores. We have our chlamydias, which have to grow within cells. We have gram-positive bacteria, like, for example, Streptococcus pyogenes, which causes strep throat. We have blue-green bacteria. We have Ricettias. The different types of bacteria can be very diverse. If we look at domain archaea, we have our methane producers. We have our halophiles, the ones that live in extreme salt. We have our thermophiles, the ones that live in extreme heat. If we look at domain eukarya, these are again going to be made of eukaryotic cells. And so within domain eukarya, we can have our four kingdom system: plants, animals, fungi, protists. And then we can keep classifying and getting more specific as we move through this hierarchy. So when we talk about our classification, these can be arranged into several descending ranks, beginning with the most general, meaning the most diversity within this category, and ending with the smallest or the most specific. So our classification in order is domain, kingdom, phylum, class, order, family, genus, species. And so the way that I remember our classification scheme is that I think of this sentence. And the sentence that I remember to help me keep this in order is I remember, do keep pots clean or family gets sick. So notice that the first letter corresponds to the classification. The do is for domain. Keep is for kingdom. Pots is for phylum, etc. One of the things that you're going to figure out is that having these mnemonic devices, these ways to help you remember things, is going to make it a lot easier on yourself. It's a lot easier to remember do keep pots clean or family gets sick than it is to remember specifically domain, kingdom, phylum, class, order, etc. So throughout the semester, you'll see that I will try and give you as many mnemonic devices or ways to remember things. I am a very visual person. I need to think about it in a way that will make sense to me and makes it easier to recall. And so I try and share those with you throughout the semester to help you remember certain things. I teach you my little tricks for how I remember these things. So if we look at our classification and go through an example, again, our most broad classification would be our domain. So if we're looking at kingdom Animalia, what domain did that have to be in? Was that in domain Bacteria, Archaea, or was it in Eukarya? Answer was the domain was Eukarya. These are made of eukaryotic cells. And so our domain is going to be, again, the most broad. So domain is Eukarya. Then we get to kingdom Animalia. So remember that within domain Eukarya, we have our plants, we have our fungi, we have our animals. So domain is very broad. Now we're getting to kingdom and we're getting more specific. So now kingdom is going to be Animalia, or the animals. So now the plants are out, the molds, mushrooms, and yeast are out, the protists are out, because those are not within kingdom Animalia. In kingdom Animalia, you can see in this example, we have our fish, we have our worm, a bear, cat, lion, butterfly, mice, right? So those are kingdom Animalia. They're still very broad, but we've already ruled out some other organisms. Then we have our phylum. Our phylum in this case is going to be Chordata. Chordates have what's called a notochord. And a notochord is a precursor to a vertebrae, essentially. And so notice that when we get into phylum Chordata, the worm is out. The butterfly is out. Those don't have that notochord. So the fish is still in play. The bear is still in play. The cats are still in play. The lion is still in play. And so is the mouse. Those all have that notochord, which can serve as a precursor for a vertebrae. We get more specific. We get our class. Class in this case is going to be Mammalia, mammals. These are organisms where the females have mammary glands to feed their young, essentially meaning they can breastfeed their young. They also have hair. So the fish is now out. It's not a mammal, but we do have our bear, we have our cat, we still have our lion and our mouse. So notice that as we go down this classification scheme, we become more and more specific. Then we get to the order. In this case, the order is Carnivora, carnivores. Now the mouse is out. Then we get down to family, getting more specific. Felidae, think of felines, the cats. So the bear's out. Then we get to genus, and the genus in this case is going to be Felis, so the felines. Those are going to be more specific to the cats. Now the lion is gone. And then we get to our species, and the organism is referred to as Felis domestica. Notice that the binomial name has the genus first, it's capitalized, species is second, it's lowercase. So our species is Felis domestica. So think of kind of like our house cats. So notice that when we classify living things, we go from the most broad to the most specific. And the most specific is going to be the species. So now we need a definition for species. So the biological definition of a species is going to be a group of individuals that have the ability to breed and produce fertile offspring. So it has to meet both of those criteria. So what does that mean? Has the ability to breed and produce fertile offspring. So for example, a donkey and a horse, if a male donkey mates with a female horse, they will give rise to what we call a mule. A mule is what we call a hybrid. It's a mixture of the two. Now, the mules are infertile. They're not able to give rise to more mules. So that's why horse and donkey are separate species. Yes, they have the ability to breed, but they don't produce fertile offspring. So they're not the same species. They're separate species. So that is kind of your definition of what a species is. Now, does that apply to everything? The answer is absolutely not. Think about organisms like bacteria. They have a species, but we can't use this term or this definition to help define a species because bacteria reproduce asexually. They simply make a copy of themselves. So we can't describe them in a way that says that they have the ability to breed. They don't actually breed at all. They simply just make a copy. And so this is kind of this overview of what a species is. But again, recognize that this doesn't apply in every instance. And similarly, if a person is infertile, meaning they're not able to have kids for whatever reason, does that mean that they're not part of the human species? No. It means that as a group, humans or Homo sapiens are of the same species, because as a group, we have the ability to breed and produce fertile offspring. So next, we're going to walk through a brief history of microbiology. And so ancestors of bacteria were the first life on Earth. So 1665, Robert Hooke, who was an Englishman, observed a thin slice of cork through a relatively crude microscope, and he reported that living things were composed of little boxes or cells, as he called them. Think of more like a cell in terms of a prison. It's like a cell. And that then led to what's referred to as the cell theory, that all living things are composed of cells and come from pre-existing cells. So he was the first one to see cells. However, the cells that he saw were not from living organisms, because cork is not alive. So he studied household objects, he studied plants, trees, and he described cellular structures and drew sketches of little structures that seemed to be alive. Now, Anton van Leeuwenhoek, 1673 and 1723, was the first to describe living microorganisms, what he called animalcules, little animals. And so what he did was he looked at different specimens like rainwater, feces, scrapings from teeth, and basically he did this using this primitive microscope that you see here. So it has this little pin where you can put the specimen. It has a lens. And so he would take those samples. He would take the rainwater or the scrapings from the teeth, etc., would put them on this pin, use this lens, and he would view those samples. And so he did these little drawings of the different things that he saw. And so what he saw was he saw these little rods, he saw little spheres, he saw a great diversity of different types of cells in these specimens that he found. And so he was the first to kind of see living microorganisms. Hooke was the one to see cells first, but his specimens weren't necessarily living. Anton van Leeuwenhoek was seeing living cells. So the way I remember this is alphabetical order. H comes before V. So Hooke came first. He was the first to see cells. Van Leeuwenhoek was the first to see living cells. So those are the people. Now, in terms of the year, I'm not going to ask you years. I will not ask you what year did Robert Hooke describe cells. That's not my point for putting these on here. The year is not important when you're studying. I just put the year on there to give you an idea of how long ago these various discoveries were made. But by no means do you need to memorize that. So once they saw cells, this led to a debate about how life arose. And so there were two competing theories during this time. One was referred to as spontaneous generation, and that is the hypothesis that living organisms arise from non-living matter, that some vital force forms life. And so in spontaneous generation, the idea was you could go from non-living to living. That was spontaneous generation. Spontaneously, some vital force, they didn't know what it was, but some vital force, they said, allowed life to come from nothing. Biogenesis, on the other hand, is the hypothesis that living organisms arise from pre-existing life. So in this case, life gives rise to life. So that life only comes from other pre-existing life. So life begets life. So life gives rise to life. So this was the theory of biogenesis. Now, there was a debate for a period of time. Some scientists believe in spontaneous generation. Others believed in biogenesis. And so we're going to talk about several experiments that were done to see if biogenesis is the way or spontaneous generation is the way. So, 1668, an Italian physician, Francesco Redi, did an experiment using meat and looking at maggots. And so what he did was he took meat and he put it in a sealed jar. And what he was looking for was did he see maggots on the meat? So if he put them in a sealed jar, did he see maggots? The answer was, when the jar was sealed, the meat was starting to break down, but there were no maggots. He repeated the experiment. He now did an open jar, so the lid is off with the decaying meat. And what he saw was that there ended up being maggots on the meat. So the maggots on the meat, basically they said this was in support of biogenesis. Life comes from life. Now, antagonists of this experiment said, well, this experiment's not valid because fresh air is required. So that vital force can't happen without air getting in. So they said that the one that was sealed, it didn't work. They didn't see life because air is required. So to squash that theory, he did a follow-up experiment, a second experiment, where instead of sealing it so no air gets in, he covered the top with a netting. So air can get in, but the flies can't get in to lay their eggs because the maggots are the larval form of the fly. So if the fly can't get in to the meat, are we going to expect to see maggots on the meat? Answer is no. Because if the flies can't get in there, they can't lay eggs and they can't form the maggots. So no maggots basically helps to suggest that it is biogenesis. Life comes from life. And so this experiment helped provide evidence in support of biogenesis. Now, it's important to note that with any scientific theory, you can't prove a scientific theory. You can provide evidence in support of a particular theory. You can provide evidence to falsify another, what we call an alternative hypothesis, etc. But we can't ever prove a theory. We can just say that the alternate hypothesis is not possible. So in this case, it's basically ruling out spontaneous generation, that life can't just come from life. So people still didn't believe that based on Redi's experiment. And so they said, well, maggots, maggots can't come from nothing. That's too advanced. That doesn't work. But things that are more simple could still do spontaneous generation. So in 1745, an Englishman by the name of John Needham boiled nutrient broth and then covered the flask. So we have this broth, this nutrient-rich media, and he boiled it to kill off any microbes. And then after boiling it, then he covered it. And what he saw was that when he looked days later, that there was bacterial growth in the flask. So the bacteria grew in that flask. So what do you think, which theory do you think that he suggested this shows? Did that show spontaneous generation or biogenesis? And his conclusion based on this experiment was that it was spontaneous generation, that life could come from nothing, right? He boiled it. He thought, well, I killed off the bacteria and then I covered it and somehow the bacteria grew. So that must be that spontaneous generation can occur, that you can get growth from nothing. However, it's simply because of a problem with the way that he designed his experiment. So if you're to think about this, if you boil it, right, and the top is still off, and then you cap it, right, then you cover it, where do you think those microbes came from? And the answer is they came from the air. They fell into the flask after the broth was boiled, right? So because of the way he designed his experimental design was flawed. And that's because he capped it after. So bacteria already had a chance to fall in after the broth was boiled. So 1765, Lazzaro Spallanzani, who was an Italian scientist, said that Needham should have covered before boiling. So to protect the microbes from the air from falling in. So he repeated this experiment. He capped it first, then he boiled it, right? So notice that nothing can fall in. What do you think his result was? What do you think he saw as his outcome? The answer is he saw no growth. So he saw no growth. That's because by covering it first and then boiling, the boiling is going to kill off any microbes. And now because it was covered before the boiling, nothing could fall in and nothing could grow. So is this experiment in support of spontaneous generation or biogenesis? Answer is it's in support of biogenesis. Life comes from life. So he argued that by doing the experiment this way, that is why you didn't see anything growing, because nothing could fall in, which shows that life only comes from life. But antagonists were still not convinced with this experiment to say that it was biogenesis. They said, well, that boiling that you did destroyed that vital force. So that's why you didn't get any growth. You didn't get any growth because you destroyed the vital force. We know that's not true, but that was what was believed at the time. So there was another experiment after this. 1861, Louis Pasteur did an experiment using this S-shaped flask. So what he did in his experiment was he took this broth and he poured it into a flask. So he took this non-sterile liquid, this broth, and poured it into the flask. Now he used a flame and he bent the glass so that it had this S-shaped neck. So notice that when he did this and he created this S-shaped flask, this is open, air can still get in, but anything that comes in is going to settle in the neck here of the tube. And is it going to be able to grow? And the answer is that if you boil that liquid, right? You're boiling that liquid, you're killing off any microbes that could be in the broth. Air can still get in. So would you expect to see growth? And the answer is that he did not see growth, right? That's because the dust and the microbes got trapped in the neck of the flask where it bent, and it never was able to get up and into the broth, and so therefore it didn't grow. And so even when these flasks sat for many, many years, still nothing grew. And so when he has his nutrient broth placed in his S-shaped flask, he heated and it wasn't sealed, he saw no growth. So is that in support of spontaneous generation or biogenesis? And that is that this evidence is in support of biogenesis. Life comes from life. The air can get in. So we can't say it's no air that's preventing the growth, right? And then he did follow-up experiments to show that in fact, that it wasn't just the act of heating the liquid that caused the microbes to not grow. It was more than that. And so let's take a look. So I have a class paper. This is going to be something you want to answer ahead of time. and then we are going to discuss it during the discussion section. So, class paper, if you break the S-shaped neck. So, Pasteur did his experiment. He boiled the liquid, right? And then if you were to break the S-shaped flask, so to break it, right? So if you break it and things can fall in, I want you to predict, would you expect to see growth? Why or why not? Right? So if you were to break it, would you expect to see growth? Why or why not? The second part of this is if you were to tip the flask over so that now the broth goes into the neck and then you put it right side up, meaning you put it back standing up, would you expect to see growth? Why or why not? So you want to think of your answer, and during the discussion, we will discuss this together. And so based on the results of these experiments and the one that we will talk about later, 1861, Louis Pasteur, microbes in the air can contaminate, but the air does not create the microbes because the air could get in. However, when he had his S-shaped flask, he did not get growth in that flask. So the air did not create the microbes. The microbes came from if they fell into the flask. And so he was credited largely with disproving spontaneous generation. Again, we can't say that he proves biogenesis, but he disproves the alternative hypothesis. So he disproved spontaneous generation. Pasteur was also credited with aseptic technique. So techniques that are used to prevent contamination by unwanted microbes. And so if you think of Pasteur, you probably think of pasteurization, right? So using heat to reduce the number of spoilage microorganisms to make milk, let's say, for example, more safe to drink. And so Pasteur really revolutionized microbiology and really started to understand this aseptic technique. How do you prevent from transferring microbes from one place to another? So question for you, which of the following statements is the best definition of biogenesis? Red, non-living matter gives rise to living organisms. Yellow, living cells can only arise from pre-existing cells. Green, a vital force is necessary for life. Blue, air is necessary for living organisms. Purple, microorganisms can be generated from non-living matter. So pause your video, think of your answer. When you're ready, push play. And so the answer is going to be yellow. Best definition of biogenesis is that living cells can only arise from pre-existing cells. So life gives rise to life. It's not red. Non-living matter can give rise to living organisms. That would be our spontaneous generation. It's not green. A vital force is necessary for life. Again, that's associated with spontaneous generation. Air is necessary for living organisms. That is not true. While some organisms like us do require air, other organisms are what we call anaerobic. They grow in the absence of air. So air is not necessary for living organisms. And purple says microorganisms can be generated from non-living matter. Again, that would be our spontaneous generation. So between 1857 and 1914, this was considered the golden age of microbiology. And so there were rapid advances in the field by both Pasteur and another scientist by the name of Robert Koch. Now that last name, Koch, can be pronounced a variety of ways. Some people will say Cook, some people will call it Koch, some people will call it Coke. It just depends on who you're talking to. There's not necessarily a right or wrong way. That name could be pronounced any of three ways. So I'm just going to go with Cook. But again, that doesn't necessarily mean the others are wrong. It just depends on how somebody pronounces it. So I'm going to say Robert Koch. Now, during this period of time, microbiology was really starting to be established as a science. So Pasteur. Pasteur wanted to know, why did beer or wine sour? He wanted to know what made it go bad. Well, yeast converts sugar to alcohol without air. That process is referred to as fermentation. So if you grow yeast in anaerobic conditions, meaning without oxygen, they will do alcoholic fermentation and they will produce alcohol. However, if that wine, for example, or beer is exposed to air, the alcohol is then converted to acetic acid by any bacteria that can get in there. So any bacteria that might be present is going to take the alcohol and convert it to acetic acid. Acetic acid, think of vinegar. It's going to make your beer or wine taste sour because of that acetic acid being produced. Pasteur demonstrated that these spoilage bacteria could be killed by heat that was not hot enough to evaporate the alcohol in the wine, but was enough to reduce the number of microbes in the wine that it wouldn't cause the wine to sour. And so this technique was then referred to as pasteurization. That's the application of a gentle heat for a short time, and the gentle heat will kill most bacteria, but it won't cause the wine to evaporate. And so he really helped to understand about, again, this aseptic technique. So what could be done to prevent contamination from unwanted microbes? 1840, we have Ignaz Semmelweis, who was a Hungarian physician. So this Hungarian physician noticed that doctors would do dissections of cadavers in the morning and then not wash their hands and would deliver babies. Because at this time, doctors were thought to be these superior people and that they were just inherently clean. And so they would do their dissections in the morning of the cadavers and then would not wash their hands and would deliver babies. And as a result, there was a high incidence of death from something called childbirth fever. However, midwives did not do dissections. They weren't involved in dissections. And when the midwives would deliver the babies, there was a lower incidence of death from this childbirth fever. And he wondered, why was it that when these midwives delivered the babies, why was the incidence of childbirth fever less than if the physicians were to deliver the babies? And so this childbirth fever results from what's called septicemia, which is bacteria in the blood. And so he noticed that the doctors had this higher incidence of this childbirth fever. And so Semmelweis concluded that some unknown cadaverous materials caused the childbirth fever. And he instituted a policy of using a solution of chlorinated lime for washing hands between autopsy work and the examination of patients. And he did this because he found that the chlorinated solution worked best to remove that putrid smell of the infected autopsy tissue, and thus perhaps destroyed this casual poisonous or contaminating cadaveric agent. And so because this chlorinated lime basically would get rid of the smell, he said, well, that likely means that it's also getting rid of this cadaverous material, which could then also reduce the risk for childbirth fever. So what happened was that he basically was somebody who really advocated for hand washing between patients. He said that doctors should wash their hands after doing autopsies. Now, people did not believe him at this time. He was ahead of his time. People did not believe him, and they thought he was crazy. He was committed to an asylum and died after being admitted by a beating by a guard. So poor Semmelweis, was telling people, wash your hands, wash your hands. And they pretty much persecuted him for this belief and said, no, physicians are clean, it's not necessary, and put him in an asylum, and then he was killed in this asylum. Well, come to find out, once they started washing their hands, the incidence of childbirth fever dropped from 35% to 1%. So Semmelweis was right. They did need to wash their hands between patients. They needed to clean their hands after working with the cadavers. 1860s, we have Joseph Lister, who was a surgeon. He applied the germ theory to medical practices, and he cleaned the instruments in between surgeries. He knew that phenyl or carbolic acid killed bacteria, and so he used that carbolic acid as a chemical control agent, basically to disinfect the surgical equipment between surgeries. 1875, Robert Koch, he was a German physician, and he came up with what's known as Koch's postulates. And these are a series of logical steps that establish whether or not an organism is pathogenic, meaning it causes disease, and which disease it causes. And he basically supported or provided evidence to show that bacteria do in fact cause disease. This led to the germ theory of disease. He discovered Bacillus anthracis from a cow that died of anthrax. So he was the one to discover this particular type of bacteria. So let's talk about how Koch's postulates work. So what happens in Koch's postulates is that he took a organism from a dead animal. So let's say he had this mouse and the mouse died, and he isolated microbes from the dead animal. So they're grown into a pure culture where there's only one type of bacteria there. They would then identify those microorganisms, so see what types of microbes were present, and then they would take that pure culture and inject it into a healthy animal. If the injected animal, the healthy animal, now dies of the same disease that the previous mouse had, and then they look to see if they culture the bacteria, the microbes from the mouse that now just died, if they then culture that and they study that, if that pathogen is the same as the initial one, then you can say that that particular microbe causes the disease, because when you injected that into a healthy mouse, that mouse then died of that same disease. So obviously that microbe must be responsible for that disease. And so there are several rules that have to be met in terms of Koch's postulates. 1, the same pathogen must be present in every case of the disease. So when you isolate from the mouse that died, the one that was injected, you have to have the same pathogen in every disease, in every case of that disease. The pathogen must be isolated from the diseased host and grown in pure culture. The pathogen from the pure culture must cause the disease when it's inoculated into a healthy, susceptible laboratory animal. So again, in step 2, they isolated the bacteria and then they inject it into a healthy host, and you're looking for, does it cause disease? And the last part of this is that the pathogen must be isolated from the inoculated animal and must be shown to be the original organism. So that's the step 5. So if you take that organism that died, the one that you inoculated, and now you take it and you look at the types of microbes that are present, if this is true, if that particular microbe causes that disease, you need to see that same microbe at the end that you saw in step 2. That would show that that microbe is the one that's responsible for causing the disease. 1796, we have Edward Jenner. Edward Jenner was basically one of the first to develop a vaccine. And one of the things that he noticed was during this time, smallpox was a very deadly disease. However, he noticed that milkmaids didn't tend to get smallpox. However, they did get cowpox from working with the cows. And when they got this cowpox, it was much milder. It didn't have the same severity that smallpox had, but those milkmaids didn't contract smallpox. And so he wondered, well, why do these milkmaids not get smallpox? And so what he then did was he scraped lesions from a milkmaid that had cowpox. So she had cowpox. He scraped some of the lesion from the cowpox and then injected that into a healthy child. And that healthy child was his own child. Can you imagine that happening now? No. Nobody's injecting my kid as an experiment. But this was a different time. So he exposed a healthy individual to the material from the cowpox lesion. And so the child then developed the cowpox, came down with a more mild form of the disease, and then exposed the child to smallpox and found that when exposed to smallpox, that the child did not contract smallpox. So something about the cowpox provided protection against the smallpox. And this protection from disease is provided by vaccination, and it's referred to as immunity, right? And if you have immunity, it means that you are protected against that disease. So think about what's going on right now in the world, right? If you think about the coronavirus, the hope is that at some point we have a vaccine, because if we can inject material from the coronavirus enough that it doesn't make us get sick with COVID, but it still causes us to mount an immune response, and that immune response then protects us from getting that disease. That's the whole idea behind the vaccination. Now, in the case of COVID, we still don't know whether or not if you get the disease, if you're even protected. So meaning even if we were to vaccinate, do you get immunity by the vaccine? Does it provide short-term protection? Does it provide long-term protection, etc.? But the idea behind a vaccination or a vaccine, vacca comes from cowpox. The idea behind a vaccine is to inject either a weakened form of that organism or just a part of that organism, just so that it elicits an immune response, but not enough so that you come down with the disease itself. And so he was largely credited with the first vaccine to be developed by doing this particular experiment. 1880s, Pasteur discovered why vaccinations work. And so he noticed that cholera bacterium lost the ability to cause disease after being grown in the laboratory for long periods. However, that bacteria was still able to induce immunity. So while that bacteria itself was no longer very virulent, if somebody came in contact with that weakened strain, that was still enough to induce immunity to protect against cholera, the disease. 1928, Alexander Fleming discovered the first antibiotic. And this has to be the most lucky discovery in the history of discoveries. This happened completely by accident. So Alexander Fleming was trying to grow bacteria, and he ended up with this contamination on his plate. And this contamination had this fuzzy appearance, and he noticed that where this fuzzy stuff was growing, bacteria was not. So something in that fuzzy material was inhibiting bacterial growth. We now know that fungus is a fungus called Penicillium. Penicillium, you can guess, produces penicillin. So this fungus is producing the penicillin, which is inhibiting microbes from growing in the area. And so it killed the Staph aureus that he was trying to grow on his plate. And therefore, this was how penicillin was discovered. It was completely on accident. He did make a good intuition where he noticed that the fuzzy stuff inhibited bacteria from growing, which then led him to further investigate. But this was a pretty lucky discovery. Now, penicillin was discovered in 1928, but it wasn't mass produced and tested clinically until approximately the 1940s. So it took quite a while for it to become kind of mainstream. So the other part that's going to be part of your class paper, meaning your assignment that will be part of the Zoom, is that you need to match the people to their contribution towards the advancement of microbiology. So like for Jenner, number 1, you're going to pick a letter that goes with Jenner's discovery. What did he do that advanced microbiology? And so you need to fill this in. And again, in class, when we discuss this, you will discuss it with a group and go over your answers. And so lastly, we're going to talk about recent advances in microbiology. So in the 1970s, we had the discovery of restriction enzymes. And so 3 scientists were the ones that are credited with the discovery of restriction enzymes. We have Daniel Nathans, we have Werner Arber, and Hamilton Smith. And they discovered these little molecular scissors inside bacteria. And these molecular scissors chop up DNA in very specific ways. And so this allows scientists to use these enzymes to cut DNA in certain ways to allow us to have that recombinant DNA technology. So meaning like when we take the human gene and we want to put it into bacteria for bacteria to make human insulin. Well, you have to have a way to get that DNA sequence into the bacteria. And so the way that that works is you use these restriction enzymes, these enzymes from bacteria, and you can use those enzymes to cut at very specific sequences. And that then allows you to mix DNA sequences together till you get that recombinant DNA, this bacterial plasmid that then has this human insulin gene on it. And so this opened the floodgates to genetic engineering and basically has helped with the investigation into biological processes and the biological revolution of the 21st century. So this was a really big discovery. In the 1980s, we have polymerase chain reaction, so PCR. PCR was a breakthrough in our ability to detect tiny amounts of DNA and then amplify them into quantities that were sufficient for studying. So you can think of PCR as essentially the DNA Xerox machine. It allows us to make many copies of those DNA sequences. This has provided a new powerful method for discovering new organisms. So it allows us to detect if a particular organism is present. So for example, in one of the coronavirus tests that we have, they use this PCR. They're looking for these sequences in the patient's body that shows that the coronavirus is present. And so they can do this by using this PCR. So diagnosing infectious diseases, so again, like for COVID, and for forensic work such as crime scene investigation. So this really basically helped for forensics because it allowed scientists to study or to basically be able to detect blood, semen, etc. that was left behind at a crime scene in very small amounts, but then be able to amplify those genetic sequences so that then they could study and figure out where did those samples come from, whose blood was it, whose semen was it, etc. That really couldn't happen well until PCR. That was kind of a game changer in forensics because it allowed scientists to amplify that DNA so that even if they didn't have a lot left behind at a crime scene, that they could still study it. In the 2000s, we have these small types of RNAs. So genome sequencing has revealed that perhaps only about 2% of our DNA actually codes for protein. So a big chunk of our DNA is not even there to code for proteins. Much RNA is ribonucleic acid, doesn't end up with a protein counterpart. And these pieces of RNA are usually small. And it now appears that they have a critical role in regulating what happens in the cell. So they control how long the mRNA hangs around, for example. And so it appears that they have a very critical role, even if they're not used to code for proteins, because normally like mRNA is used to code for proteins. But these other small RNAs, even though they don't code for protein, they still have a very important role. And this has led to new approaches in how diseases are treated. For example, if the small RNAs are important in bacteria that infect humans, they could then be new targets for antimicrobial therapies, because these small RNAs, what we call small interfering RNAs, which are siRNAs, or miRNAs, which stands for microRNAs, if we have those small RNAs that target a particular bacterial sequence, that could be used to then help treat diseases. And then we have the Microbiome Project. And so this was done in the 2010s and beyond. And so the first detailed information that was produced by the Human Microbiome Project was astounding. Even though the exact types of microbes found in and on different people are highly diverse, the overall set of metabolic capabilities the bacterial communities possess is remarkably similar among those people. So even if they had a unique type of bacteria in their body, and you compared two people, right, person A and person B, they might have different bacteria between person A and person B, but those bacteria that are present are doing something very similarly for the body. And so this and other groundbreaking discoveries have set the stage for new knowledge of our microbial guests and their role in the overall health and disease. And so an interesting example of this was that they found that babies who were born via C-section, so cesarean section, versus babies that were born vaginally had very different types of bacteria as part of their normal flora. They were influenced by the way they were delivered, right? Because babies that come out of the vaginal canal are exposed to a very different subset of bacteria than babies that are cut out of the uterus, right? They're not coming in contact with those vaginal bacteria. And they're actually finding that that vaginal bacteria is actually good for babies to help colonize and be part of their normal flora. And so one thing that they're doing now is to basically do transfers of that, vaginal area onto babies that are born via C-section to try and recreate that natural normal flora that they should have if they had come out vaginally. There are lots of examples of this. We can have what are called fecal transplants. So for people who have a type of infection in the gut called C. diff, Clostridium difficile, this is a type of bacteria that is very resistant. And one of the ways that we can treat this is by a fecal transplant, by transferring feces from a healthy individual into the intestine of somebody who has C. diff. And the idea is that when you put those good bacteria in, that they will outcompete the bad bacteria. And that could be used to then fight off this C. diff infection. And so a lot of research is going into this Human Microbiome Project, understanding what bacteria are present in the body, what happens if you get disruption to that normal flora, how do you get these different diseases, like which bacteria are associated with obesity, right? Which ones are associated with obsessive compulsive disease, etc. And so this is part of that human microbiome project. And so this concludes our first lecture.